Good morning, everyone. Good afternoon. Welcome to the second day for this workshop. We are going to kick off with a panel discussion. We are fortunate to have four participants from four different regulatory agencies. And this panel discussion will try to address some challenges in the development and validation of PVVM and safe space from regulatory perspective. But just before we start, I'd like to give you some a couple of housekeeping remarks. You may ask questions via the QA tab on your dashboard at any time during this presentation. And at the end, we try to answer those questions. And if you need technical assistance, please use the chat. So let's go to this. I think it kind of gets frozen at the beginning. See it happen again. All right, here we go. To set the stage, I like to spend some few minutes reminding you about a current issue, which is the need for more affordable medicines. And as you know, regulatory agencies such as the FDA are taking new steps to modernize how sponsors and the agency can rely on tools and analysis. And this includes a more widespread of model and simulation, as you heard yesterday from our keynote speech given by Dr. Madabushi from the FDA. I think there is a delay with this. Okay. FDA believes that model integrated drug product development, in other words, the combination of models and data, will likely result in higher confidence on bioequivalence, in the assessment of bioequivalence, and efficacy and safety at earlier time in drug product development, which might reduce the number of studies needed, thus decreasing the cost of medicines. The recent publication of the guidance on the use of physiologically based pharmacokinetic analysis and biopharmaceutics applications, which I will refer in this presentation and panel discussion as PVVM, demonstrates that the agency, the FDA's initiative towards supporting more affordable medicine is real, right? As you may know, this guidance provides a good overview on model development and validation, as well as modeling applications. But like with any other guidance following onset of application or publication, the application of this has generated several questions and the need for further clarification. So one of the goals of this panel discussion is to raise or reiterate awareness of issues related to building and validation and application of these models. And as much as possible, we'll try to address these issues. So what are those issues that I'm talking about? Well, first of all, just let's make sure that we are all on the same page when it comes to what is PVVM and what are the difference between PVVM and PBPK. Now, PVVM needs or starts with a PBPK model. This means that before building a PVVM, a PBPK model needs to be built and validated using the typical inputs as we are familiar with. However, PVVM goes beyond PBPK in terms of additional data requirements and modeling steps, which are focused on creating an in vitro and in vivo link between the critical material attributes, process parameters, dissolution, and systemic exposure. A key difference is that in PVVM, an IVIVR or IVIVC need to be developed, which will allow for the establishment of safe space. And the advantage of PBBM is that it may not be confined to building an IVIVC offering a simpler path to biowaivers, especially for immediate release drug products for which, as you may know, the rate of success of IVIVC is rather low. So, as I mentioned before, building a PVVM starts with the construction and validation of a PBBM baseline. But I mentioned that it requires additional data, which is focused in gathering additional dissolution data from formulation variants and the corresponding CP time profiles. But the question is here, how many formulation variants do we need? And how critical is it to include a non-BE batch? Also, how critical is to have a biopredicted dissolution method? Can one still develop a safe space with a method that is, for example, over discriminating? And if so, what are the limitations of such a decision? Now, when it comes to integrating the data into the platform and building the model, the guidance, the PVBM guidance emphasizes the importance of incorporating dissolution into the model. And as you may know, based on publications, there are many different approaches that we can utilize to achieve this goal. So the question is here, which approach should I use? And how do one assesses the predicted performance of this to determine which is the best approach? And when it comes to uh, the development of the IVIVR and IVIVC, which one should one utilize? For example, for an extended release formulation, is this sufficient to build an IVIVR, or do I really need, I must build an IVIVC? When it comes to the validation, then which criteria should I use? Should I also use the same criteria for IVIVR versus IVIVC? And so once we have reached that, then obviously we one can build the safe space. And how do we build that safe space? But so first of all, what is safe space? Is the boundaries defined by dissolution profiles within which drug product variants around the target are anticipated to be bioequivalent to one another. And just to explain a little further this concept, let's assume that we have 3 formulation variants around the target, and these observed of in vitro and corresponding CB time profiles that I don't have here are known as knowledge space. So let's assume that when these formulations are tested in vivo, the pink, the red, and blue are found to be bioequivalent, but not the green profile. So then a safe space can be defined based on the extremes of dissolution profiles that were bioequivalent. And this safe space, or this space is called dissolution safe space, in a way that any future major changes can be justified without additional B studies as long as these profiles falls within this safe space. Now, the question that comes over and over, and perhaps very critical here, that I see that we must address is, in the absence of non-B data, so let's assume we don't have this profile here, in which circumstances is it possible to extrapolate beyond this knowledge space? Now, with that in mind, let's go into the panel discussion. And as I mentioned to you, we are very fortunate to have 4 participants that do not need introduction because they are very well known for all the contributions to the agencies in their fields. And we have Susan Cole from the Medicines and Healthcare Product Regulatory Agency in the UK. We have Gustavo Mendes Lima Santos from the Agência Nacional de Vigilância Sanitária in Brazil, Anvisa. Hopefully I didn't mispronounce that much. I see that you're smiling. Good. We have Shereni Berangingham, sorry for mispronouncing, from Health Canada. And we have Lian Sao from the Office of Generic Drugs, FDA. So the first question is, what could be the largest single change that would cause an upsurge in the use and acceptance of PVVM and PBPK model and simulation in support of drug product quality biowaivers. And I like to invite, let's say, Lian to provide his input on this question. Lian?
Hey, Sandra, can you hear me okay?
Yes, thank you,
Okay,
Lian. Welcome.
great. Yeah, it's my pleasure to share some thought with the audience, with the community, and with the other agencies. Uh, Before we open up, I think, PBBM and, the guidance you're referring to, Sandra, has reminded me all the discussions we have had, including you, during the guidance development within FDA. I think it's a good guidance, and also with lots of in practice, still need as you mentioned, still need many clarifications. Since modeling is a novel field, and in the practice, there's no doubt there would be some uncertainties in terms of the concept and applications. Concept-wise, I still think PBBM is kind of a subset of PBPK modeling, rather not to confuse the field. I think it's a controversial issue. I know someone is holding a different opinion, but that's my understanding for now. Regarding your question, what will be the change that can cause an upsurge in the use and acceptance of PVPK modeling to understand drug absorption and to support biowaiver? I would say drug product quality biowaiver, something confusing me. I would think you are referring to the Invariable B study biowaiver. When using the PBPK, PBBM to support the risk assessment of the impact of food, excipients, gastric pH, and special populations, formulation change on general, I think that those will be hot topics pretty soon. I'm telling this because the topics are related to the SCH M13 discussion points and the revised PKB guidance. The FDA just revised guidance last year. You can see in the draft there are lots of new changes in terms of biowaiver. In many occasions, you may resort to PPK modeling to support the biowaiver and to support other regulatory need as well. In that regard, I think the SCH initiative and the revised PKB guidance will serve as not a change at the event that could trigger loss of upsurge of applications in PPK model in terms of understanding drug absorption. I think that you will see the change in the next 2 to 3 years. Thank you very much.
Thank you so much, Liang, for your input. And I just want to say, yes, there is a controversial debate in terms of how to refer to this PVPK application to drug product quality.
Mm-hmm.
You read the title of the guidance, just long title. So I think that PVVM just switches very well from the perspective of different people. But again, no matter what we call it, right? That this is not, as you know, we are not discussing in this how to call it. It's just to address the issues behind it. I would say certain gaps in knowledge that need to be addressed. So with that, I'd like to invite Sue to give an input on this, please.
Hi, yes, you can hear me okay, can you?
Yes.
Yes. So, yeah, unfortunately, I can't think of a single change that would have that sort of big impact. But really, to reflect what Liang was saying, I think it will come down to increasing experience and visibility, really, as we see more examples. And experience in the UK in regulatory submissions is quite limited at the moment. But, there are a number of groups within the US and in Europe really looking at this methodology. There's a new IQ group being formed. And in addition to sessions like this, I think, this is really increasing the visibility and also giving that learning on the methodology. So, so I think, it'll be nice to see, the new examples coming through, and nice to see a number of really well developed and validated, well publicized examples, maybe some with robust IVI receives. And I think that would be really, give some strength to this area. So I think the sort of workflows that are being discussed by some of the groups, and like the one that you showed, would be really useful, and maybe to see, to standardize it more. But yeah, so that was just my thoughts on this.
Great.
Thank you.
Just to follow up on your comments, I wonder if it is possible just to briefly talk about this group that is being formed by the IQ. Can you talk about it, or is it still, is it confidential at
I
this
don't
moment?
think so. So, as far as I know, there is this group being formed with under the remit of IQ. So, and regulators and a number of industries are involved. And, and it will be to look at examples of PBBM modelling and, really to get the regulators' input on it. So I know the FDA are involved, EMA, Brazil as well, so most of the regulatory agencies. So, it will be going through some examples and getting the feedback on that. So yeah, as far as I know, that group, is being set up at the moment, and I don't think it's confidential. I hope it's not.
Great. And so just so because I think this would be a great thing to have, right? If I may
Yeah.
ask an additional question on that. Is this the purpose of this IQ group is to, could you please just elaborate a little bit more, is to better understand, to fill in the gaps into this approach, the application of PBPK in support of drug product quality, which we call PVVM for short?
I'm not sure I'm really the best person to give a lot of detail on this group, actually, but my understanding is to go through some work examples and see what the regulators would be looking for, maybe in terms of the verification and validation of models. So, but yeah, I'd be a bit uncomfortable really commenting any further on
We will,
that.
in due time, we will know. Thank you so much, Sue, for your input. Gustavo, may I invite you to provide your input on this question, please?
Yes, good morning, good afternoon, everyone. Thank you, Sandra. Thank you, all my colleagues here, to give us the opportunity to share some of the perspectives here of Anvisa. And it's quite good to see that we are going through this path of having these tools used here in Brazil in our submissions. And I believe that my input here would be echoing my colleagues saying that as the experience we have as regulators here in Brazil shows, when we don't have a very harmonized road, a harmonized guide that bring us the workflow as Sandra showed here, and bring us the what we call here in Brazil regulatory safety to take actions. This definitely brings the idea a little more hesitating in the sponsor perspective. So I believe the change. I believe it's going to be a big change to have this discussion on international forums like ICH, and it was mentioned here the M13 that is giving a very good work on considering this kind of approaches. And I believe that also considering the situation where Sandra mentioned, cost of development is really something, and the pandemic situation also provided more and more difficulties on this. So I believe that this also is a very good and important point to bring a change on this scenario regarding the submissions here in Brazil.
Great. Just a brief follow-up, Gustavo. Does Anvisa have any plans? I don't know. Obviously, it would be for everybody here except FDA. Does Anvisa have a plans for implementing a guidance for PBBM?
Yeah, definitely. We are right now with a lot of initiatives here in Brazil of training our team and also writing a guidance that consider the experience we have with the examples we have, and also considering the international discussions and the scientific discussions. I cannot say exact dates for this, and we know how regulatory works happened with a lot of time, a lot of discussion. But definitely, this is something that we are going through, and this is something that we believe that in next periods we'll have another good news.
Thank you, everyone. Let's move to the second questions we have chance to. Okay. Second question. When building a PVVM model and a safe space for immediate release drug products, what are the data requirements for dissolution and PK? How critical is this the incorporation of a non-bioequivalence batch? And let me invite Jereni to take the first stab on this.
Okay, thank you, Sandra. And hello, everyone. It's really a pleasure to be here and to be able to discuss improvements that we've seen over the last couple of years in this field. So data requirements for a PBBM and a safe space for immediate release drug products is an evolving topic. At Health Canada, we've seen an increase in submissions for PBBM specifically for immediate release products over the last few years. And also there have been recent journal articles on this topic, including case studies from industry and SIMS Plus collaboration that was published last year. And also the recent guidance from the FDA has also helped to clarify the situation around this. In general, immediate release solid oral dosage forms that contain a highly soluble drug substance are considered to be low risk with respect to the impact of dissolution on in vivo performance. A PVBM approach then would be constrained to drug substances that are more low solubility, so BCS type class 2 and 4 drugs. And the best practices for data requirements are still evolving. So for immediate release drug products, dissolution should generally be determined under discriminatory conditions so that you're able to establish a link between product dissolution and the relevant quality attribute. And for immediate release products, this would very often be the drug substance particle size. Formulation variants of different or differing release rates and quality attributes are required to assess the sensitivity of the dissolution method over the intended range. For the absorption model, you'd need dissolution inputs from at least 2 formulation variants with corresponding concentration time profiles for oral administration. And these profiles, the concentration time profiles, should exhibit the same rank order as the dissolution profiles. In addition, for the disposition model to describe the PBPK, you would need PK profiles following IV administration of the drug. To define the safe space, the range of virtual disposition profiles should contain the target, which would be the bio batch or a pivotal clinical batch, and that batch's dissolution profile. So while a PBBM can be defined for regulatory purposes with 2 formulations, additional data sets of dissolution and corresponding PK profiles for validation would increase the confidence in the modeling outcome, and that would likely enable a greater range of flexibility from a regulatory perspective. The last question on the slide is, how critical is the incorporation of a non-bioequivalent batch? So this would be considered a best practice. So a batch that's non-bioequivalent would be defined as a batch for which the 90% confidence interval of the test to reference geometric mean ratio of the AUC or the ratio for the Cmax falls outside the 80 to 125%. So for Health Canada, for the Cmax specifically, we'd use a ratio and not the 90% confidence interval for the majority of drug products, which would mean drug products that are not critical dose drugs. So including such a batch, which is non-bioequivalent, would actually test the model's ability to reject batches that are not bioequivalent. And this is really important to be able to challenge the model. And this would increase also the knowledge space and facilitate being able to design an appropriate safe space. So I think that's my thoughts on this so far.
Very comprehensive. Thank you so much, Rennie. And I might follow up with another question, but let's give the opportunity to give their input to Gustavo. Please, do you want to follow?
I'm just it was a very comprehensive answer, Srini, and I just think to contribute the challenge that us regulators have to have access to non-BE batches to challenge the model, this kind of things, because what right now happens is that we receive the batch with the results with the best development aspects. The development process of formulation for early development and also for generics happens to be presented only with the best. And it is a change of culture here in Brazil, at least in my perspective, to convince sponsor to share results of non-bioequivalence batches and other dissolution to establish this knowledge space with better, a more comprehensive aid. I'm not sure if this challenge is also for the other regulators, but here is something that I'd like to point out.
So maybe I can add to that, Gustavo, that in our experience, when we've seen it, it hasn't been a full pilot batch that's non-bioequivalent, but it's been a formulation in development phase where they've had a batch that has failed, and the sponsor has PK data for it. And that would be adequate in my mind for this kind of purpose.
So do you want to comment on that?
On this question?
Yes, on this question,
yeah.
Yes.
So I mean, I agree. I think Shirini gave a really comprehensive answer in terms of the data requirements for dissolution and for PK. So, and really, if you're going to extrapolate outside of the different absorption profiles that you've got, then that non-BE batch really defines your boundaries. So, I would agree as well that that's important, and we should encourage people to submit that data. So yeah, wasn't really anything that I could add really in terms of the answers that are already been given.
Good. Leanne, not sure if you want to comment on this.
Sure.
Or
yeah, I agree with all the colleagues have shared. I want to make clear, to have a safe space for the immediate dissolution profile is not required to have a non-BE batch, but it's good, very good to have one. Similar with other applications, the development model need to have sufficient purpose-dependent validation. I think the key is whether we have sufficient dissolution data and PK data to develop and validate the model based on the purpose. We have a publication actually in the pediatric realm, how to extrapolate the dissolution space from adult to pediatrics. In that paper, I can share the reference if there's any interest. We actually built the model with more than 10 clinical data set with their corresponding dissolution profiles, and this paper is good to set dissolution safe space for Tamiflu for pediatrics. So if you have interest, I can share the reference with everyone. It set a routine I think not necessarily use a non-BE batch. It can get data from other formulations and PK data from other formulation with the same API or even different APIs with similar natures. That's my share for this topic.
Right. Thank you so much, Lian. So you mentioned, Lian, just to follow up on that you build a safe space using information from different populations and different
Formulations
yeah,
as well.
different drug products, formulation and populations.
Mhm.
So in your mind, do you think that's possible to build a safe space to support drug product quality using data from different dosage forms and different populations, or do you think this safe space is specific for that drug product, meaning that you must have formulation variants from that product to build that safe space?
Yeah, I think the purpose using data from different formulations to again to challenge the model. You can use non-BE batch to challenge the model, but it will be case by case. And if for certain products, if we bad luck have many data available already, so to sufficiently validate the model, so why would we bother recommending another non-BE batch? But however, if there's no such data available, and if the B space is really, really critical for the product itself, and if there's a lack of in vivo data, then probably you need a non-BE batch to challenge the model. And many times I just feel in the regulatory agency, we may not, I don't think it's a routine to have a safe space for high dissolution profile. I think it's depending on the regulatory need, right? I hope, the other, uh, depending, the safe space could serve two purposes. One is for product quality purpose, the other for BE. For BE evaluation, then I think you need to have very a good model that can predict the in vivo performance of the product based on the bio-predicted dissolution profile. It's very challenging. I don't think there are many good successful cases. There's still room for us to invest in that area.
Right. Thank you, Lian. So if I were to summarize what you said, just to make sure, at least in my mind, is clear. So it would be correct to say that to build a robust PBPK baseline, one could use data from all kinds of formulations and populations just to make sure that the predictive ability of that baseline model is confirmed, right? But when it comes to fit for purpose, when you want to determine whether a change, just like we have a fit for purpose model for DDI, you need clinical data to validate that path, right? So in this case, PBBM is another fit for purpose modeling approach. And to make sure that it's able to do what it's supposed to do, which is to make sure that the model is going to pick up a difference in formulation and it's based on the knowledge of or is based on the concept of IVIVC and IVIVR. So that means that we need formulation variants around the target for that dosage form to be able to have to waive those bioequivalent studies after a formulation change was implemented. So hopefully that uh, that would summarize somehow the applicability of PBPK versus this PBPK for drug product quality.
Yes,
But
Sandra. Yeah, I think you summarized very well. Yeah.
thank you so much, Lian. So let's go to the third question. I see this is a tricky question, but let's give it a try. What metrics and criteria should we use for judging the predicted performance of PVVM IVIVR and PBBM IVIVC, would the same criteria apply for highly variable drugs? And you know that the PVVM guidance, please, I'm going to refer to the recent guidance on PVVK PVVM as I mentioned before, that this has a clear description on what would be the criteria for validating an IVIVC, and that borrows from the 1997 guidance on the IVIVC, right? And so, but based on the application of these models and after the publication of the guidance, do we still believe that that criteria should hold true as per the guidance? And I'd like to invite Sue to address this, take the first stab on this question, please, Sue.
Okay, yes, so this is very much of interest to me, really. So how we judge the predictive performance of the model, is it done on comparing the curves of the observed versus the predicted, or based on PK parameters, and then what statistical criteria? And we, in our PBPK guideline, we didn't put in any criteria, really, because we considered it dependent on the application and to some extent on the drug itself. But you have to remember our PBPK guideline covers all aspects of PBPK, from drug interactions, special populations, and includes the biopharm as well. So I have found some criteria in our modified release guideline. So we have quite an extensive appendix there which discusses IVIVCs. So really there we say that the IVC model is generally accepted as adequately accurate if the visual inspection of the entire concentration curve is well predicted and the prediction errors are within acceptable limits. So we say internal predictability is assessed using the IVIVC model to predict the concentration time profile from the respective dissolution data for each formulation. And then the summary parameters, Cmax, AUC, etc., are calculated from the predicted concentration curve and compared to the respective parameters for the observed. And here we give some numbers. So we say the absolute value of the prediction error for all summary parameters should be less than 15% for each formulation. And the average prediction error for all formulations included in the IVIVC development should be less than 10%. And I think this 10% figure fits in with the one that Sandra was referring to as well from the FDA guidance. So, but I also know that this 10% figure may seem quite strict, and there has been some controversy around this. So, and particularly as Sandra mentioned, highly variable drugs, can we, really expect those to come within that 10%, or, could we have some flexibility around these? So yes, I've just put that out there as a proposal, but I'd be really interested to hear other people's views.
Thank you so much, Sue. I know this is challenging. Liang.
Yeah, sure, Sandra.
Thank you.
I think Sue already covered the base really well. I don't really have much to add. But I think currently the IVC is mainly based on the secondary PK parameter like Cmax, AUC. I think in the future, as you labeled as PBBM, IVR, PBBM, IVC, I will call them mechanistic IVC, IVR, should be the direction to go. It can definitely offer us more insight about the in vitro, in vivo correlations. I also agree with Sue mentioned that the 10% of in terms of the PE percentage to qualify an IVIVC is pretty strict. I think in practice, well, depending on the case, we should apply some flexibility on that. Guidance is guidance, but we have alternative to guidance as well. Regarding highly variable drug, given the nature, if you apply the drug, give the drug to the same individual twice, multiple times, even from the same individual, you observe in that regard, certainly present a challenge to establish the AV-AVC relationship. In this case, probably more AV-AVR if we can master the trend by a mechanistic understanding. Certainly, at the end, everything boils down on the regulatory use of the model. So that's my share to this question.
Thank you so much, Lian. Jereni, just wanna, if you were to address this question also, Lian, and I think also Sue mentioned that it will be case by case to perhaps expand those criteria. If you could please, in your answer, address that in what kind of tools, what additional information may be needed to expand these criteria.
Okay, so
thank you.
At Health Canada, we don't have a guidance. So everything, as far as we're assessing models, has been case by case, taking into account international guidance that's available. One of the things that's different for us, as I mentioned before, is our Cmax parameter. For drug products that are not clinical dose drugs, we assess our bioequivalence for the Cmax based on ratio of the test to reference product. We recognize that the Cmax parameter is a little bit more variable in general compared to the AUC, and therefore we tend to show a little bit more flexibility with criteria applied to the Cmax. I agree with my colleagues that the 10% on the mean has been difficult to meet for a number of the submissions that we've reviewed. I feel that the 15% prediction error is a reasonable point to be at this time, especially as we move towards physiologically based IV-IVCs and IV-IVRs. Once again, I think there'll have to be a lot more discussion internationally so that regulators are on the same page. And maybe the initiative that Sue mentioned, exactly. The IQ collaboration with industry will help towards this, because the aim there is to review case studies and for the regulators to have a discussion while reviewing it.
Great.
So that may help with that.
Thank you so much, Sereni, for your input. I think you nailed it. And I think that this effort that is being led by IQ is going to really help us to better address that issue. Just in the interest of time, let's go to the next question. Question 4. Under which scenarios could one consider extrapolating outside the knowledge space in order to expand the limits of drug specifications? And what data would be required to increase confidence in or support the extrapolation? And somehow Lian already addressed it a little bit, but let's hear from Gustavo. Gustavo, you want to take the first stab on this?
Yes. So in our perspective, in our internal discussions, extrapolating outside the knowledge space, building the knowledge space is challenging based in the information we have received from the development of sponsor right now. And extrapolating outside that, of course, would be something that it should be based on data on different formulation variants. So the effort that the company has to do prior to the submission of the regulatory agency, identifying the critical parameters for variants and showing us how can we build this knowledge space, and how can we expand based on the relation or the correlation of IV-IV is something that we would like to be convinced of. So I cannot say one specific scenario, but I know that we have to be convinced that the data show us this confidence, and this kind of data is the data based in the results of the dissolution, different dissolution profiles, establishing PBPK, and then how it's set to the target. So this is something that is pretty much in our perspective a convincing step, a convincing path for us to accept.
Thank you, Gustavo. Sereni, you mentioned several times about risk assessment. biopharmaceutical risk assessment. Could you, when I invite you to answer this question, also consider that when
Sure.
answering your question? Thank you.
So I would say that if a safe space is defined using a conventional bracketing approach, which reflects limits that are defined by bioequivalent formulation or process variants, in that case, our relationship is considered to be qualitative. And I don't think there's really a place for extrapolating outside of that. So I take a very conservative approach on that and would not really support extrapolation in that case. If you have a PBBM approach and a mechanistic understanding of the in vivo drug release, absorption, and PK profiles, perhaps, and really it's perhaps, there's a place for extrapolation. At this point, I don't think there's sufficient confidence, but we've taken a totality of evidence approach sometimes, where we've considered evidence outside of just the PBBM model. One case could be when we have extra exposure response analysis data. I think, Sandra, I've heard you talk about a situation that you've had this in. And we take a similar kind of approach where extrapolation really needs to be supported by linear pharmacokinetics, uncomplicated drug, low-risk drug profiles, such as a BCS class 1 product. It's a number of factors that would be considered in the risk assessment. And at this time, therefore, extrapolation outside the knowledge space is really a very confined and not a very common occurrence, I would say.
Great, thank you. Thank you, Sereni. So, may I invite you to give your input on this?
So I think, like the others, I would be very cautious about this. But also, you know, as Shirini said, I wouldn't say it's a definite no, though. You know, there may be circumstances. So, you know, if you have got a very good, well-established, well-validated IVIVC, then maybe some extrapolation would be allowed, you know, outside of that. But I think it would be limited, you know, and you would be extrapolating, I suppose, to say this would still be within, you know, the bioequivalence, rather than extrapolating to say, well, it's not bioequivalent, but, you know, and then your exposure response understanding comes in. So it really depends on the circumstances. So yes, I would be cautious about going outside of the space that you've established, but you know, I think in some cases, you know, if it's a minor extrapolation, maybe that could be accepted.
Great, thank you. And we had already input from Leanne from the previous question. So let's go to the next question. Question number 5. When defining the boundaries of a safe space, should the upper and lower boundaries be compared to one another, or should the upper boundary should be compared to the target, or the lower boundary to the target? And so it is always a confusion there. And even when I was at the FDA, we had some circumstances where, depending on the data, right, depending on whether it was exposure response data, we could do it. But it's still not clear, and usually guidance don't have recommendation level of detail. But over and over, this question comes, and I like to invite, let's say, the Sereni to take the first attempt to answer this question, give their input.
Sure. So my understanding is that an intent of a safe space is that all formulations with dissolution profiles or other relevant quality attributes within that space, so all formulations with these characteristics, are bioequivalent to each other. So to achieve this, the predicted performance at the upper and lower boundaries should be compared to each other. And in general, a maximum difference of 20% or less is in the predicted means of the AUC and Cmax is considered adequate to ensure safe space, where formulations at the boundary limits are considered to be bioequivalent. For a generic product, we also expect that formulations at the upper and lower boundaries of the safe space are bioequivalent to the reference product. But as you've mentioned, Sandra, there are cases where we would consider a comparison of the upper or lower boundary to the target batch. And this could be appropriate, like you mentioned, when there's other evidence to support it. This is generally, in my mind, an expansion of the safe space, right? So when it's supported by adequate exposure response data to ensure that there would be no impact on safety and efficacy, that could be possible. So in general, this approach would be considered within the context of the totality of the evidence and not necessarily based on the model itself.
Great, thank you, Sereni. Gustavo, please.
I think I agree. I will echo Shireen. So totality of evidence may lead us to accept the comparison, the upper boundary or lower boundary to target. Comparing upper and lower boundary to one another is the way we think that can bring us this bioequivalence area that we would accept. But in many cases, I believe that it could be the best way to consider case by case the totality of evidence to address this question.
Thank you, Gustavo. Leanne, may I invite you to provide your input
Yeah,
on this
sure.
question?
I agree with the comment from colleagues, and I think it's more like a philosophical question rather than a scientific question. So if we think about the in vivo studies, we are not compare, say, if we are doing generics, one generic to the other generics, we are compare one generic to the reference. Could it be the exposure could it be relatively low for one of the generic? The other one could be relatively higher. I think we haven't been expecting, the generic should be bioequivalent to all of the other generics in the past. I think it's kind of a sensitive question, but we have to consider from that angle as well. In terms of the boundaries, it's more like either we want to be more stricter with the in vitro testing results or more accommodating. Either way, to me, have some justifications. I think at the end is totality evidence. If we want to rely more on the in vitro data for, with the minimal in vivo data support, we may want to apply more strict criteria. Otherwise, if there's in vivo data, you are doing a B in vivo B anyway. So why would you be so strict in the in vitro testing? The release may be less or more biopredictive. I think at the end, the in vivo data will trump, will tell you whether the drug is really bioequivalent or not. That's my input.
Thank you so much, Leanne. Let's move to another question. So that, by the way, we have put together 10 questions. Not sure if we're going to get to all of them, but let's try. Let's try maybe one more or two. Number 6. What metrics and criteria should one use for judging the appropriate approach for incorporation of dissolution data into the model and platform? And I'd like to invite Liang to take the first step on this.
Yeah, sure, Sandra. I think it's a good question. Actually, for this question, we have two modeling communities, right? The pharmacometrics and the biopharmaceutics in this area. For the model validation verification in pharmacometrics, if you're using non-map, you want to model the clinical response, then you are using the visual predictive check, quantitative predictive checks. And for the dissolution data, we are talking about the endpoint, the output is PK. They were talking about whether you can predict Cmax or AUC deeper, whether you can predict the shape of the PK curve. In that regard, I think currently we have a prediction error at the quantity to measure the goodness of fit. Other than that, I really don't have good input. I think it's more users' choice, modelers' choice. At the end, I don't think FDA has very firm recommendation on how to what the exact quantity you want to use to justify the model. I think it still stays as of a state of art for the modelers.
Thank you, Liang. Let's see, Gustavo, do you want to provide input on this?
Yeah, I wouldn't. As we don't have the guidance and the criteria defined here, I would just echo what Liam said, and I think that it is a question that definitely have to be considered of different perspective, the modeling and also the biopharmaceutics. When we think about the incorporation of dissolution in the model, and we know that as we have planned the idea of PVBM and constructing modeling, definitely is something that we have to take to have in mind and use with the best. There's different ways of inputting this solution in the modeling is something really important to consider, because depending on the way, you have some bias that may make the model not applicable.
Thank you. Gustavo, just to follow up a little bit, because I think this is a very important topic and perhaps it would be the last question that we address. Let's say that somebody decides to input the data as it is. What would you say? What would you tell the sponsor? What justification do they need to say, yes, I'm going to input the dissolution data as it is into the model? Let's say, Liang, what would you say on that?
Yeah, I think, Sandra, depending on the purpose. So I can use a case example to let everyone appreciate where we are struggling. So we have approved a product, it's a topical product, diclofenac, topical gel. And if we use a purely bottom-up approach, right, the model, the PBBM model didn't predict the PK profile in an ideal manner. I think it stays true for every case. So even with the same dissolution profile, if you are same product, if you are conducting multiple in vivo studies, there will be study-to-study variation. And that will be, if you feed data to one particular study very well, you will fail to predict the PK data from another study. That is very likely the case. I think in this case, we need in the bioequivalence assessment, the purpose is not for us to predict a perfect PK curve. I think the purpose for us is to evaluate whether the two formulations are bioequivalent. In that case, we actually did a sensitivity analysis. Say if we by optimizing one parameter in the model, we have a perfect fit to the PK data set or to any PK data set you have obtained. Then with the different input from the different formulations, then you evaluate whether they are bioequivalent. Then it's not only for one PK profile, then you keep changing all the parameters, then you evaluate it under all the scenarios, the two formulations, the two dissolution profiles at the input will always get the equivalent output. In that case, you are pretty sure, you gain confidence that these two formulations, the two dissolution profiles predict bioequivalence. Then in that case, I think we can make a regulatory decision making. Actually, we were heavily criticized by, people in our publication, you see the bottom-up approach didn't predict the PK concentration in an ideal manner. how would FDA hold such a loose standard? Actually, there are other thinkings behind that. Given the totality evidence, there are a good battery of in vitro data testing, not only dissolution, plus other Q3 parameter comparisons. We made the decision.
Great.
I'll stop
Thank
here.
you so much. We are at top of the hour, so unfortunately, time's up. Thank you all for joining us. A special thanks to all the panelists for the great and valuable input. At the end of this track, there will be a group of Q&A session, including perspectives from all track speakers to address any unanswered questions. Please make sure that you stop by, and the session is over, and hopefully you're able to continue joining this workshop. Thank you again, everyone, for your great contributions. It was my pleasure to be part of this discussion with you. Bye-bye.
Thank you, everyone.
Bye.
Thank you.
Bye. Bye.
Bye.